This is the second video talking about bryophytes. So overview of this video, we're going to look at lineages of bryophytes, and we're only going to look at two. We're going to look at Anthocerotophyta, the hornworts, and Marchantiophyta, the liverworts. If you have maybe a not super recent botany textbook, even recent botany textbooks might refer to the liverworts as the Hepatophyta, which hepata for liver. So that name was recently changed to the Marchantiophyta. So our bryophytes were the first group of plants to evolve on land, we think, and this says that they are paraphyletic. That's currently unknown. The research that I've been using to kind of evolve our botany course actually says that they are likely monophyletic, which goes against pretty much anything else you'll read. And so that just means that they would have had a single bryophyte ancestor that then led to their lineage where you diversify from there and we get these different groups of bryophytes. Whereas if they're paraphyletic, we sort of get one group of bryophytes branching off and we stay on this sort of path toward the rest of plants. Then another group of bryophytes branches off. And so it's just about, can we group them all and trace them all back to one shared common ancestor that's only shared by that group, or do they sort of share ancestors with other groups along the way? And it's looking like from this 1,000 transcriptomes project that they might be monophyletic. So there's three major lineages. All of them are small and prostrate, and so there's exceptions to this, but that they lay kind of flat to the surface or aren't able to hold themselves upright because they lack lignified vascular tissue. So when we learned about xylem, we learned that it's made of elements, vessel elements, and tracheids, and those are lignified cells with that secondary cell wall. Bryophytes don't have that. So because of this, they require wet conditions to survive. They don't have roots to penetrate down into some kind of soil surface. There isn't a soil surface for them to penetrate down into, right? We just have this rocky terrestrial surface when they first move. So they're going to be reliant on, their fungal associates to help them scavenge water, and maybe reliant on their cyanobacterial associates to help them fix nitrogen, so they have access to nutrients. So they require wet conditions to survive. I'm getting on a little sidetrack here. Except a few mosses, especially there's large groups of mosses now that can survive in incredibly dry conditions that most other organisms can't. They have the ability to completely dry out and then stay in that condition and then rehydrate later and live, which most organisms can't do. So mosses do have some kind of exceptional abilities to be dry, even though, as a general rule, bryophytes need water. They do need water to reproduce because their sperm have to travel through water to fertilize eggs. All of the bryophytes also have gametophyte dominance. So when you look at a bryophyte, you see this green tissue. It's the largest part of the organism. It is haploid. So all other plants, when we see this larger part of their life cycle, that's the green photosynthetic part, that's a sporophyte. But for bryophytes, it's the gametophyte. So this is a kind of trait that unites them together. And so the gametophyte is the larger part of the life cycle. It generally has a longer lifespan and it's nutritionally independent. So it's the photosynthetic part and can feed itself. So there's always exceptions. This is Dawsonia superba. It is a moss that can grow up to 2 feet tall. It does not have lignified vascular tissue like we see in plants, but it has this sort of analogous set of structures where it has hydroids and leptoids, where it's able to kind of transmit water and sugars from its upper parts to its lower parts, but it's not lignified. Let's look at this first group of bryophytes, our hornworts. And current research that I'm using says that it's likely that the hornworts are not genetically closest to vascular plants, but perhaps they evolved first. So I'm just going to cross that off because who knows right now? It's all up for debate. So what they look like is this flat kind of flap of tissue that has little root-like structures. So this whole thing is the gametophyte. These little root-like structures are called rhizoids. So that just means root-like. They don't function to absorb water. They don't have any vascular tissue within them. They just function for anchorage. So they are to kind of hook themselves to the terrestrial surface. And you do find fungal associates with these rhizoids. So they sort of had this possibly mycorrhizal relationship pre-roots that allowed them to maybe access any nutrients in that kind of rocky surface and help break down that surface. So this is our thallus. And in this thallus, for hornworts, often you'll see little pockets where they have cyanobacteria living inside them. So again, that's another association that would have allowed them to access nutrients in this kind of harsh landscape. And this is why we call them thalloid, is because they're always this sort of flat flap of undifferentiated tissue. And on that thallus, you'll have these little kind of vase-like structures, and that's where the egg would have been produced inside this archegonium, and so the sporophyte grows out of the gametophyte. So here's our sporophyte. This is all diploid, and it is one big sporangium. And unlike most plants that we'll see, there's only, I think, one other example of this that we'll see during our botany class. This sporophyte grows from a basal meristem. So we're used to seeing apical meristems, right? The shoot apical meristem and the root apical meristem, this tip-oriented growth where new cells would normally be produced at the tip. However, in this sporophyte, new cells are produced at the base, and it pushes the rest of the sporophyte growth upward. So the oldest part of the sporophyte is up here. Ooh, also interesting that the stomata and cuticle are formed on the sporophyte, but not necessarily on the gametophytes. So we're going to start seeing a lot of adaptations that are beneficial for living on land happening in the sporophytes, and that's actually going to lead us to this sporophyte dominance. Another feature that helps you recognize hornworts, if we're going to zoom in on this here. So if we're looking at a hornwort under the microscope, its cells are going to each have one large plastid with a pyrenoid. And those pyrenoids are sort of ancestral features. We see pyrenoids in our green algae, like in our Spirogyra. And so that is maybe pointing toward a more ancestral trait. So here we see our thalloid liverworts, or not liverworts, hornworts. So this is the thallus. It's this flap of undifferentiated tissue. It's growing pressed to the surface, prostrate, and then it has the sporophytes growing up out of the gametophyte. So that's that matratrophy, where the embryo is nourished and retained, and then it's fed through a system of cells here by the gametophyte. And then here on the right, we can see the monoplastidic condition where we have one large plastid inside each cell. So that's pretty unique, and it's a good way to tell apart a thalloid liverwort from a thalloid hornwort. Here we see the hornwort sporophytes that are maturing and releasing their spores. So they start to dehiss from the top down, because this is where the most mature spores would be. And they can dry out because they have stomata all over them, and those stomata allow the escape of moisture to dry out the sporangium. And once it's dry and those spores have matured, it will dehiss, so it'll split into these different segments that kind of twist. And there's little elaters in there, these little kind of fibrils that help disperse the spores, and it does that from the tip downward, from the most mature part to the least mature. Our liverworts can look like either hornworts or mosses. It makes them a little tricky. They can reproduce asexually by structures called gemmy, and they have simple pores for gas exchange. So unlike the hornwort sporophyte that had stomata, which would be regulated by guard cells, liverworts are just going to have holes in the gametophyte that can't open and close, so they can't regulate their gas exchange. And here it says most ancestral lineage, maybe not. We don't know. there's two different kind of body plans for a hornwort, not for a liverwort. Sorry, I've had to re-record this like 4 times because PowerPoint keeps quitting on me. So now it's all just word salad. Okay, so here's our leafy liverwort. It has these kind of major ranks of leaves. The larger upper leaves are going to be produced in this sort of flat sheet. And then if you flip it over, you'll have these ranks of little underleaves that are smaller and stick out of the bottom. And mosses don't have that. So if you're trying to tell the difference between a leafy liverwort and a moss, you can look for those underleaves. They're really small, but you can still see them. They'll produce rhizoids along this surface. At any point they want to sort of attach to that surface, and they can produce the sporophytes either along the body, or some species will produce them just at the end of these structures. So these are all sporophytes, but now they're divided into 2 main parts. We have the seta, which is the stalk, and we have the sporangium, and that's where meiosis happens and spores are produced. That sporangium will generally dehyst open, so when it dries out, and split into these kind of 4 compartments. And so that helps release the spores. And we will look at the thalloid liverworts when we draw the life cycle. So here you can see the leafy liverwort and these ranks of leaves. So you have these larger upper leaves that would be on the upper surface. If we were, this is flipped over under the microscope. We could look at it the other way and it would look just like a moss, except for a few key features. But the underside has these little tiny leaves, so make sure to look for those. This is a picture of what that looks like in a hopefully less confusing way. Here is our thalloid liverwort, and this is one that you might see invading a nursery or greenhouse or anywhere where it's kind of like shaded, cool conditions where there may be growing shade-tolerant plants. This is a good invader, and it invaded the terrestrial surface, so it's not surprising. So over here on the upper right, we can see an archegoniaphore. So the archegonia would be produced under these little branches right there, and then the egg would be produced within the archegonium, get fertilized, and develop into a sporophyte, which we're seeing these really early sporophytes developing here. They look like coconuts under a palm tree. And then the Anthocerotophyta, instead of being that tree-like structure, it looks more like a flat plate, and that's because water is going to bounce onto the Anthocerotophyta and splash the sperm out of those Anthocerotophyta and hopefully onto an archegoniaphore. So here we see those archegoniaphores growing on a thallus, and that thallus has all these little dots. Those are simple pores. And it also, if we look over here on the bottom right, we might see these structures. These are those gemmy cups, and they don't have very many gemmy in them anymore. They've probably all bounced out and been splashed out by water. But it's just a little disc of haploid tissue that is going to be a clone of the gametophyte that can land somewhere and grow into a new thallus. So we're going to draw the Marchantia life cycle and label all the parts. So meiosis is over here. Random line is over there. Realistically, I should leave myself more room on top, but I did not, so here we go. So let's start with some spores. So we have spores that are produced by meiosis. They're going to be genetically different from each other. Some of these might grow into a male gametophyte that's going to produce anthocerotophytes, while others might grow into a female gametophyte that's going to produce archegoniaphores. And then both could produce gemmy. And those gemmy would then just grow into another gametophyte of the same type, right? So gemmy from a male gametophyte would grow into another male gametophyte. Gemmy from a female gametophyte would grow into another female gametophyte. So we have the archegoniophore. So that just means the structure that bears the archegonia. And here we have an anthocerotophyta. So the structure that bears the anthocerotophyta. These are the gemmy cups. Gemmy cup with gemmy. A singular gemmy or singular gemma is called gemma. So here's our gametophytes. We're going to zoom in on the archegoniophore and the anthocerotophyta. So if we look underneath a branch of that archegoniophore, there's going to be archegonia, kind of nestled into the kind of area where those branches come out. And inside that archegonium, there'll be an egg. So let's label this. We have the egg, which was produced by mitosis. And this structure here is the archegonium. And this is the neck or the venter of the archegonium. If we zoom in on the anthocerotophyta, we would see on the top of that structure, oh no, I'm drawing this wrong. They're embedded in that surface. Let's start this again. So we have this sort of disc-like structure that's at the top of the anthocerotophyta. That's the antheridial head, and kind of embedded in that are all of the antheridia. And so this is where sperm will be produced. So those little sperm are going to get splashed off of the antheridial head and then travel to an egg. When we get the egg and the sperm meeting, we get fertilization, which you might see in some of your textbooks or resources called syngamy. That's when you're thinking of karyogamy and fertilization both happening. Okay, so fertilization. This happens on the female gametophyte in that archegonium. So this is the archegoniophore. And that's the archegonium, and then now we have the zygote, which forms on that female gametophyte. That zygote is going to grow by mitosis. And it does that growth on the female gametophyte. So here's our archegoniophore. And it has sporangia with seta that grow underneath it. So each of these purple structures is a sporophyte. And each one has a seta, it's that short little stalk there, and a sporangium. And meiosis happens within that sporangium to make spores. Here's a picture of some leafy liverwort sporophytes. They are very cute. I love finding these. They are pretty ephemeral. They don't stick around for long. So here's some that get produced at the end of that gametophyte. Right, so here's our seta, the stalk, and the sporangium at the top where the spores are produced. So that's the site of meiosis. For all plants, this site of meiosis is going to be inside that sporangium. So here we see they have opened by drying out and push themselves open. That's dehiscence. So they've dehissed to release their spores. And this looks a lot like a moss, right? You might think it was a moss, but you'd have to take a close look at those leaves or look for these sporangia.